Now let's talk about gene expression, right? DNA carries genes. We daughter cells just inherited this whole genome from their parents. Now they need to decide what cells to become. Is it to become neurons or become muscles or become right? Yeah, skin cells. They need to come together, form tissues, form organs, make up a form a whole human being, right? Functional human being. How will they do that? They need to express, right, the genes that they carry. So what's the first step in gene expression? How do I express the information coded in a gene in DNA? What's the first step in expressing that? In determining the shape of our nose, what do we do first? We need to convert the gene to what? Or the DNA to what? To RNA. Right. That's the first thing we do, right? We can what is our nose made up of? Proteins, right? Yeah. Teeth, shape of our face, everything, right? Our organs, they are all made up of proteins, enzymes in the form of enzymes, in the form of structural proteins, right? Extracellular matrix, yeah, connective tissue, proteins. The code for those proteins is where? In the DNA, the genes. So each protein has a gene, technically. Although I can make different types of the same protein from the same gene, but each protein should have a gene, which means each protein is unique, right? So, but we cannot convert the genes directly to protein, right? How easy would that be for a medical student, right? I can just go from gene to protein, right? I don't need to learn some intermediate step in the middle, right? Less information. So we can't do that. So we need to convert it first to what? RNA. Yeah, convert it first to RNA to a form we can read or decode. So we cannot decode DNA directly. So what's the process of converting DNA to RNA? That's all called what? Transcription. Correct? Transcribing DNA. Taking DNA and converting it to another molecule. Yeah. Transcription. And what enzyme will carry out transcription? Thank you very much. RNA polymerase. RNA polymerase synthesizes RNA from DNA. So the first step is I need to, of course, the first thing is RNA polymerase. It can bind to DNA, like, but the problem is it has a low at some certain points in DNA. It has a low affinity for DNA. Now it's gonna bind to DNA and then fall off. Binds to DNA, falls off. DNA falls off, right? So it doesn't bind very long to DNA molecule until it gets to a point, right, where it has a very high affinity for DNA molecule, where it can bind and stay. And that point is what we call the promoter region. The point on DNA where RNA polymerase has the highest affinity for DNA molecule is what we call the promoter site. And promoters are usually what? They are usually just proximal, right, to the DNA of interest. So each gene basically, sorry, to the gene of interest, each gene has a promoter. Some genes have multiple, sometimes a promoter can regulate, right, multiple genes, but a gene must have a promoter region. And that promoter site is usually upstream to the gene, usually upstream. So remember, we want to read. We always read from 3 to 5 prime. It doesn't matter. Replication transition, we're going to read from 3 to 5 prime. So we can say the promoter will be on the 3 prime end, just before the gene, right? Yeah, of interest. So once DNA polymerase gets to that promoter region, it's going to bind to DNA, right, with very, very affinity and stay. And promoter region is made up of what? It's made up of Tata box, TAT, and CAT box. So Tata and CAT, which means it's a region that is abundant in what? Adenine and thymine, which makes sense. It makes sense because it's going to be easier to open up that part of DNA, right? Does that make sense? Compared to if the region is rich in what? Guanosine and cytosine. Triple bonds. It's going to be way harder. So Tata box, CAT box, they form the promoter region, which represents where RNA polymerase has the highest affinity for DNA molecule, and it also marks the origin, right? Location where we're going to open up the DNA, right, to begin transcription. So once RNA polymerase binds to the promoter region, together with general transcription factors, we call them general transcription factors because they are not specific for any gene. They play a role in the transcription of all the genes in the body. So they are general. So RNA polymerase binds to that promoter region together with some general transcription factors, factors 1, 2, 3, etc., which will allow RNA polymerase bind tightly, right? Open up the DNA at that point and will start, right? Transcription. So RNA polymerase will transcribe, right? The gene transcribe the gene to RNA. Now, the problem with that, right, is that process is slow. It's slower. It goes at a basal rate, right? And that is why in the body, right, every gene is always transcribed unless the gene is turned off, like methylation, right? It's heterochromatin. If the gene is euchromatin, it's always be made. It will always be transcribed at a basal rate. That is why every protein in a cell, right? There's no protein in a cell that is zero. In healthy people, every protein that a cell is supposed to make, right, or every gene that is euchromatin will be making their protein. So you will never find absence of the protein of a gene, right, in the cell. You never see that. You never see it. Never be zero. It will never be zero. Never. So there's always a basal level, right, of every enzyme in the cell, which means every pathway technically, right, is taking place always in a cell at a basal level. So right now, every one of us is going through every pathway in the body is active at the basal level to some point. But we need, right, a way to regulate those pathways. We need to be able to make the pathways faster at some point or make it even slower at other points, almost turn it off. And that's why that's what we call gene regulation now. So how do we regulate the speed of transcription? That's why you have what? Specific transcription factors. Where in addition to having general transcription factor, each gene has its own specific transcription factor. And those specific transcription factors, they don't bind at the promoter site. Where do they bind? They bind at what? Enhancer or repressor sites, depending on if you want to increase the speed of transcription or slow it down further. Yeah. Enhancer and repressor sites. That's where they bind to. Which can be located where? Anywhere, right? In relation to the gene. Yeah. Unlike promoters, they are always upstream to the gene. Enhancer repressors can be anywhere, sometimes even within the gene of interest. Sometimes before the gene, sometimes after the gene. Yeah. So when specific factors bind to the enhancer site, they need a second signal, which is something to activate them. And that's why we have our hormones and signaling molecules. Remember those hormones we said are steroid hormones and steroid-like molecules that can enter the nucleus? Yeah, they bind to those what specific, right, transcription factors, and they can turn on certain genes or turn off certain genes, right, depending on the state of the body. Exactly. Thought ties together. Yep. You are literally learning, right? From the creator, right? Of the universe. That's why medicine is so beautiful. Yeah. You're learning from God directly. Well, if you believe in creation and not evolution, whether there's a big bang or small bang, there's somebody somewhere at the beginning, right? Of it all. Yeah. Everything just makes too much sense, right? To be by accident. And if it's by accident, then why don't you find humans anywhere else in the universe? We should have met aliens by now. Yeah, accident happens more than once. But if something is designed, it's gonna be unique and it's gonna happen. It can happen once, right? And once alone. Yeah. So, so special generation factors, right? So you want to increase the speed of it. So although, let me give you an example. Although gluconeogenesis is taking place at a basal rate, no matter what our metabolic state is. But now, when I'm fasting, the pathway will be what? Faster. Because now I have what? Cortisol, glucagon, epinephrine through cyclic AMP, and cortisol directly entering this nucleus, binds to the specific transcription factor for gluconogenic enzyme genes, increase the speed of transcription, make more enzymes, and increase right the speed of the what? Of the pathway at that point. That's how you regulate biochemical processes as well as a lot of things right in the body. Yeah. All right. So I transcribed RNA. What RNA will I make first? We make what? hnRNA, right? Heteronuclear RNA first, which is RNA that contains what? Introns and exons, right? Because not every part of a gene will code for a protein. Some sequences in the gene are what? They are nonsense, rubbish. They are non-coding sequences. Some sequences in the gene of a DNA, they are what? Non-coding. They don't code for proteins or amino acids. So those are the ones we call what? Introns, right? Introns. The exons expressed, the ones that are expressed, that codes for proteins are the word exons, the coding sequences. So when I initially transcribe DNA to RNA, I'm going to transcribe both the introns and exons, correct? And I have what? I have an hnRNA, heteronuclear RNA. So what do you do next to that hnRNA? Splice. You splice the hnRNA. And what does splicing entail? Remove the introns and you join what? The exons together. And once we do that, we'll now form what? Messenger, right? RNA. And all these are happening where? We're still in the nucleus, right? We haven't left the nucleus yet. All these are still happening in the nucleus, right? All these are happening in the nucleus. What splices hnRNA? What splices? What is the name of the enzyme that splices hnRNA? Spliceosome, right? Right, spliceosome. Spliceosome, right. Yeah. Spliceosome splices messenger RNA. And spliceosome is a combination of what? Of ribonucleoproteins and small nuclear, right? RNAs. The small nuclear RNAs combines with ribonucleoproteins. We call them SNRPs. right? They form spliceosomes. Yeah. And there's a spliceosome can recognize splice sites, right, on the hnRNA. Splice sites. Typically, the splice site is actually like I think GT rather GT sequence. Yeah, guanosine type, guanosine and thymine GT sequences. They form the splice site, and the spliceosome should be able to recognize those sites, and so that it can correctly remove the introns and join the exons together. So there are some diseases that result from what? Splice site mutations. Where the mutation is at the splice site, which means spliceosome cannot recognize the splice site. So what will it do? It's going to include introns, right, in the messenger RNA. So what happens to the protein that will be made? The protein that will be larger, right, bigger than normal. It's going to include amino acids that don't code, that shouldn't be there. And what happens to the protein? The protein will not fold properly. And it's not going to work. Classic example, beta thalassemia. Yeah. Some cases of beta thalassemia are due to what? Splice site mutation. Yeah. So we formed the messenger RNA, or while forming messenger RNA, what else do we do? What other modifications will RNA messenger RNA undergo? No, before translation. Where do you translate? Where? Is it in the nucleus or cytoplasm? In the cytoplasm, right? But where are we still? We are still where? In the nucleus, right? So what must we do? We must get messenger RNA out, right? Of the nucleus, transport it into where? Cytoplasm, right? Yeah. But what's the problem with that? There's a lot of problem with that, right? One of the problem with that is that what? We have enzymes that can degrade nucleic acids. We have nucleases, endonucleases, exonucleases that can break down, right? Nucleic acids. So you need a way to protect, right? Messenger RNA. Secondly, we need a way to what? To add a signal to messenger RNA to allow it to leave, to exit, right, the nucleus. So that's why you have what? 5 prime cap. You had 17, right? Methylguanosine cap to the 5 prime end. And then you do what? You had poly-A tail, multiple adenosine, right? To the tail, 3 prime end of messenger RNA. Yeah. We're adding a cap on the 5 prime end of messenger RNA. We call it 7 methylguanosine cap. 7 methylguanosine. You're going to add it to the head, basically, of messenger RNA. Then at the tail, you had what? Adenosine, multiple adenosine molecules. We call that poly-A tail at the 3 prime end. There are three reasons why we need to do that. First, I already mentioned two. First, the 5 prime cap is the signal for exiting the nucleus. They allow messenger RNA to leave the nucleus. Then both the 5 prime cap and the poly-A tail will protect the head and the tail of messenger RNA, the 5 prime end and the 3 prime end, from degradation, respectively. And then finally, their presence is necessary for initiating translation. Their presence is both of them must be there, because that's the only way for ribosome to know that what that the messenger RNA is complete and intact before I start translating it. So if it's not intact or complete, then I don't need to waste my time right translating now. You just degrade it, yeah, and wait for the next messenger RNA. Yeah. So get it out, exit, protect it from degradation as well as initiating, right? Translation. That's why we need those right molecules. So finally, the messenger RNA leaves the nucleus, makes its way to the what? To the cytoplasm. So what do we do next? Now we're ready for what? Translation, right? Is it every messenger RNA that is made that makes its way to cytoplasm that will be translated? Is it all of them? No. So that level is another level of regulating the gene. So remember, we're always regulating from the beginning, from the level of DNA. We're regulating. Methylation, acetylation, heterochromatin, euchromatin. We are controlling which genes will be expressed, which ones are not. From the level of the splicing too, we have regulation there. Sometimes you can combine different exons. You have alternative splicing sites to make different types of proteins. Yeah. And then we get to the 5 prime, 3 prime cap. Yeah, that is also a level of regulation. Because if we don't have them, the messenger RNA is degraded, we're not expressing the gene. But if we have them, we get it to the cytoplasm. Now, when you get to cytoplasm before translation itself, you can still turn off messenger RNA. That's why we have silencing RNA, microRNAs. That's their job. We have silencing RNAs and microRNAs. Their job is to what? They can bind to messenger RNA and turn it off, prevent it from being translated. That's another level of regulating gene expression. Then you have P bodies in the cytoplasm. There's a structure called P bodies, which is basically where you have all these microRNAs, silencing RNAs that will bind to messenger RNA, right? And kind of turn it off, keep it quiet until I'm ready to translate it. Or sometimes even degrade it completely, right? If you don't want to translate it at all. Yeah. That's another level of regulating, right? What genes are expressed and which ones are not. So assuming we want to translate that messenger RNA, then the next thing is, right, we have our players, tRNA, right, to bring in the amino acids and read the what? The codons, right? The codons on messenger RNA. Then we have ribosomes, the large subunits and the smaller subunits, yeah, that will come together with tRNA and form what they're going to form, and the messenger RNA, they're going to form what? A translation initiation complex. Yeah. And then once they form that complex, right, we can start translating that messenger RNA into what? To protein, which is and protein is what? Is a polymer of amino acids, basically. Yeah. So, and I talked about sorting yesterday, right, already, of proteins, where all those proteins we're making, right, in cytoplasm, they have different targets. Some of them are going to the mitochondria. Some of them ends up in where? In the peroxisome. Some remain in the cytoplasm. Some are to be excreted out of the body. Some are for the cell membrane. Some are for the lysosomes, right? So you need to sort. How do we sort? They all have what? Signals, unique signals, right? That will target them, right? To different location where they need to be. And for those that are meant for the lysosomes, cell membrane, and outside the cell, extracellular matrix or space, they have the hydrophobic signal, right? We call what? Signal peptide, which will be recognized by what? SRP, right? On the RER, and they're going to be translated where? Into the what? RER for proper folding, from there to the Golgi, and from there to their proper target. And then for those with lysosome, they need what? A second signal minus 6-phosphate, right? To get them to the what? Lysosomes here. So we've gone what? Full cycle. Questions? Mm-hmm. Right, regulating generation at the level of the messenger RNA. Remember, messenger RNA is basically the gene. We've taken the gene from DNA, converted to what? HNRNA, right? HNRNA to messenger